Thanks, Missy. Thanks very much. Thanks for coming. So I'm Paul. So I work here in Whitechapel, as you said, between the Royal London Hospital, which you will have seen on the way in, the big blue building, where I work as a trauma surgeon, and then the rest of my time is here, where I'm sort of trying to study and understand how injury responses develop. So I spend most of my professional time thinking about injury in some way, and that's really the crux of what we're going to talk about today. So injuries are a fact of life. They affect all of us, and if you're anything like me, these types of injury happen pretty often. They're things that we need to be able to overcome to carry on with our daily life. And actually, with or without this sort of melodramatic, histrionic reaction that sometimes ensues. And any of you who are lucky or unlucky enough to spend time around toddlers know that even from the first, as soon as we get going, injuries are just part and parcel of what we have to deal with every day. So it's very important that for us to live normal lives, we are able to respond and recover from injuries appropriately. And this is actually quite a good depiction of how we classify trauma into blunt injuries on the right. So this is, for example, stubbing your toe, catching your finger in a door, something like that, where there isn't actually necessarily a wound, as opposed to penetrating trauma, which usually involves a sharp implement and a wound. And these are actually quite different conditions, and we'll sort of talk about that a bit later on. So, of course, not all injuries are minor, and I think in all probability most people in the room have in some way been impacted by severe injury, either themselves or by knowing someone who's been affected or even died from a serious injury, and I know that I have. And this is a really important public health problem. It's a really important and common cause of premature death, and so it's really important that we actually understand how people respond to injuries so that we can support recovery. Just to put that in context with some facts and figures just briefly, so this is from the Global Burden of Disease study. It's a little bit old, and essentially what this global analysis shows is about 10% of the total global burden of disease is attributable to injury. So these are disability adjusted life years, which is like a standard metric of quantifying how much a disease is impacting an individual. And you can see that injuries are right up there with what we would think of as the biggest sort of health issues in our society. And I'm going to use the term trauma from now on throughout this talk, which I'm using to mean physical injury, the disease of physical injury. And we very much think of this as a disease. It has all of the ologies that you would associate with any other disease, epidemiology, so it has particular risk factors. It has a specific pathophysiology or biology, how it actually affects the body, which is what we're going to talk about. And we can make prognoses just like with any other condition. So they're not just things that happen to people, I guess is the overall message. We think that this is actually best understood as a disease, just like the other conditions that you see on this list. This is just another depiction of that same information. So basically here, the non-communicable diseases are coloured in blue. So you've got ischemic heart disease, which is heart attacks in the top right, and cancers up on the right there. The red are the infectious diseases. You'll see a few that are obviously quite familiar. And then the green here is injuries. So just to depict the scale of the problem at a global level. Again, this is in terms of disability adjusted life years, so a combination of premature death and also premature disability. So it's a really big problem for us to understand. So that's the context. This talk is really about how the body actually responds to injury, and hopefully I've kind of convinced you that it's an important thing that we should try to understand, both for our just everyday lives and also for this important clinical problem. So I think before we get to the question of how the body actually responds, the first question really is, how does the body actually know it's been injured? How do we actually sense injury internally? This is really the question that I think a lot of us have been working on for a while now. It's actually relatively recent science, I think surprisingly, given how important this problem is. So I thought I'd start by just showing you this picture of what the response to injury actually looks like. So what you're looking at here, the red is actually a thermal injury, so a burn to the liver. And the green dots are neutrophils, which are sort of the white blood cells, and they're the sort of first responders of the immune system. And this is live imaging of the liver after a burn. And what you can see is those white blood cells are migrating into the burn, clustering around it quite rapidly, and then starting to get rid of the damaged tissue and initiate the process of repair. So this is from a publication in 2010, which I think is a really nice depiction of an immune response that is happening in the absence of infection. It's completely sterile. And to me, it reminds me a lot of the classic video that many of you will have seen of a white blood cell chasing down a bacteria and then eating it, which, if you haven't seen it, have a look on YouTube. It's a sort of classic example of how white cells follow molecular cues and chase the bug and then basically eat it. And to me, this is just the same. These white cells are following some sort of molecular signal that drives them in towards this injured area, and then they can start dealing with it. And this, I think, is the key concept, that you have an injury to a cell, and that can be, a mechanical injury, like you catch your finger in a door, or a burn. There's some sort of signal, and we'll talk about what that signal actually is, and then you get a response. You get an immune response to that injury. And this sort of construct, this simplified construct, is basically how we respond to injury. And it's a relatively new concept. The idea that you are responding to damage, not to an infection, not to a virus or a bacteria. You're actually responding to damage. This is Polly Matzinger, who is a famous American immunologist who put forward in the mid-90s, so not ancient history, well, semi-ancient history, this idea of, she called it the danger model. She's a very interesting person to look up. She's notable for many things, including co-authoring a paper with her dog, which actually got her banned from submitting to the Journal of Experimental Medicine once they found out. But lots of other interesting stories about her if you want to Google her. But basically she put forward this idea, which was quite controversial at the time, perhaps still is, that actually the immune system is not primarily interested in self or non-self, which is the prevailing paradigm. In other words, the immune system is not sensing something that is not from your body, like a bacteria. It's actually sensing damage. And her argument is that when you have a cell that's in trouble, stressed for whatever reason, it says to the immune system, I am being damaged, not I am being confronted with something foreign. And I think this is a really elegant construct for thinking about injury responses. It also explains lots of other sort of peculiarities of the immune system. For example, the example that Polly Matzinger used was pregnancy, where there is a foreign organism that the immune system is completely able to tolerate without mounting a reaction. Whereas a transplant, which is a foreign thing, is often rejected, which she argues is because of damage. So it's quite a complicated model, but I think for what we're talking about, it's a really useful idea. And this is essentially how the injury response develops. So what are these damage signals, basically, is the next question. How does the immune system actually pick up on injury? So we term these signals damage-associated molecular patterns, or DAMPs. They were called alarmins, which I actually think is a better name, but seems to have dropped off. So we now call them DAMPs. And basically, the idea is that anything that disrupts your cell or stresses out a cell in the body will cause stuff that is inside the cell to go outside the cell. And then that is a signal to the immune system that something is going wrong. And actually, it may also be that if something is in the wrong place inside the cell as well, not just getting out. But definitely, this is, I think, the simplest way to think about it. When I was trying to think of an example of or an analogy, for some reason, I don't know if this is a very good analogy, but came up with this idea of Jelly Baby. So the Jelly Baby packet is the cell, and then So normally all the jelly babies are inside, and then if it's open, the jelly babies come out, and that is a sign that something has happened that you need to deal with. I get the jelly babies away from the children usually who can eat them all up. And obviously these different coloured jelly babies can represent different molecules. I'm not sure if it really helps, but anyway, jelly babies. So what are those things inside the cell that actually trigger a response, trigger a reaction? This is actually a really interesting field, I think. It's thrown up a lot of really interesting discoveries. So this is your sort of standard schematic of the cell with all the stuff that's inside the cell. And normal conditions, most of these things should not be outside. It's just the normal thing is they're inside. As soon as they're outside, it's a sign that something's gone wrong. So what are those things that the body detects? Well, the first one that actually provoked a lot of attention was the mitochondria, which many of you will know of as the energy producing unit of the cell. So it basically makes energy for the cell. It's thought that these mitochondria actually, in ancient evolutionary history, were bacteria that the evolving cell basically brought inside it to use it to generate energy. So the mitochondria is quite interesting because it's got its own DNA that is actually quite similar to bacterial DNA. It's not like our DNA in certain ways. So essentially these researchers were looking at injured patients and seeing that they basically looked like they had an infection, even though they'd only just been badly injured. And they thought they would look in the blood to see if there was any evidence of these mitochondrial components. And basically, so this is a classic paper, again, it's about 15 years old now, where they've measured mitochondrial DNA in the blood of injured patients and compared it to healthy volunteers. And essentially they find that it's massively increased within the first few hours after injury. So that there's somehow the mitochondria is getting out into the blood. And what they show in this paper is that mitochondrial DNA is actually a really powerful activator of neutrophils in the way that those green dots that I showed you going into the burn. And this video wasn't working, unfortunately. I'll leave it up to see if it does start working. But basically, they've got a bit of this mitochondrial DNA on the pipette tip, which is that arrow. And if the video worked, you would see the neutrophils going and chasing after it, just like the white blood cell chases after bacteria, and just like those cells go in. So your own mitochondria, when they're in the wrong place, trigger this really robust inflammatory immune reaction that basically, as I'll tell you a bit later, can be harmful if you have too much of it. But certainly we think represents a fundamental way in which you recover from injury. So that's one example. The other example of something inside the cell that might be driving this is actually in the nucleus. So the nucleus is where your DNA is housed, and I had to cast my mind a little bit back to this, but when I first started studying this. But basically, your DNA in our cells it's not just naked; it's wrapped around these proteins called histones. So your chromosomes they're basically a combination of DNA and histone. So you have loads of this stuff in the cell. It's one of the most abundant proteins in all cells. And so in the cell, histones are absolutely crucial for regulating how your genes are expressed. It's really kind of one of the most fundamental aspects of cell biology. But it actually turns out when they're outside the cell, they also act as signals that damage has occurred, which again, it kind of makes sense. Like, they shouldn't be outside. When they are outside, the immune system seems to be able to detect that actually they are a signal of damage. And we were very interested in these partly because there's so much histone in the cell. So we were thinking, of all of the things that are coming out, we should probably start by going for the things that there's a lot of. But they have these other interesting properties. So one of them is that some cells will actually, so neutrophils again, these attack cells of the immune system, actually deliberately push out their nuclear material in these, they're called extracellular traps or nets. So they will actually when they're activated, will actively release histones and DNA into the bloodstream to kind of trap bacteria, we think. And as shown up here, these dying cells will also release histones. So this is another way that the immune system is signaling that it's been injured. Again, if you measure this in the blood, this is actually our data from patients at the Royal London Hospital. These are blood samples taken about an hour after injury, and each dot here is a patient, basically. So on the y-axis is how much histone they've got in the blood. You can see that basically the severely injured patients, the trauma column, have got loads of this histone floating around in the bloodstream. I would add that, just to clarify, these are patients who've had serious injuries. These are not patients who've caught their finger in a door. And we'll talk about how those things are perhaps distinguished, because I think if you do just stub your toe, you probably will have a bit of cellular damage, histone release. As what I'm going to get to in a bit is, what does it look like in a patient who's had a really massive injury? And actually what you see is that cellular leakage spills over into the whole bloodstream and causes responses that aren't just targeted like you saw in that video, but they're actually going all over the body. And that's when we think things get problematic. Just to also show you, this is the same or similar cohort over time. This is something that happens very quickly. So you have a big release of histone into the blood at time 0, which is about an hour. And then by 24 hours, by 72 hours, it's pretty much gone away. So something's happening really quickly that then fades away. Studying trauma is quite, in terms of understanding responses, we know exactly when the thing happened. So with infections, when you're studying patients with infections, you never know when that infection started. It's quite tricky to get to that. And patients will present at all different time points. With trauma, you know exactly when it's happened. And so looking at responses over time is actually really quite informative. Fine, so we'll come back to the histones. But basically, this is our model now. So you've got cellular damage, release of these damage-associated molecular patterns, and then you get a response. So the next part is obviously what's the response? How does the body actually sense the release of these signals? And the answer is that your immune cells have specialized receptors, so proteins on the surface, that are actually designed to detect these damaged molecules. And these are called pattern recognition receptors. There's lots of different types. The most famous type are called toll-like receptors. And these receptors were actually first discovered in fruit flies. They're actually discovered because they did something completely different in fruit flies. But the point is that they're absolutely ancient in evolutionary terms. They go way, way back in our evolution. And so basically we've evolved these receptors to some extent respond to damage. Now, interestingly, there's a lot of overlap between the response to damage and the response to infection. And some of these receptors will actually recognize bacterial components, and they will also recognize damage molecules from just a damaged cell. But I think the fact that these DAMPs signal through these receptors that are so ancient is a good example of how important it is that we've evolved a response. And to me, this just makes sense that we must have evolved to respond to injury, because it's just so important for, as I just said at the beginning, how we go about our day-to-day lives. One example of this concept of how important it is in evolution that I came across relatively recently is actually from an Attenborough documentary, which was called Kingdoms, about different species. Anyway, essentially there's one story, and I just found this quite instructive as an example of why it's so important to be able to have a good response to injury. So it's about this chimp who is the alpha chimp. He's running the show. He's living his best life. But he's getting a bit old. And then essentially he gets attacked by a bunch of other chimps who want to take over, essentially. And they really badly beat him. He's really badly injured. He loses a digit. He's got lots of wounds all over his body and things. And the documentary sort of follows what he does over the next few days. He's basically static for a bit. They sort of leave him for dead. He gradually starts resuming activities. He does survive. He starts drinking a bit of water, eventually gets back to some food. And eventually he does come back and sort of takes over the group of monkeys again and reestablishes himself as the alpha, and then has the opportunity to pass on his genetic information again. So it's just quite, it's not particularly complicated story, but I think it really, whenever I watch these now, I'm just thinking in terms of, most of these wildlife documentaries involve some sort of injury or threat of injury, and being able to respond and recover from that, I think, is just absolutely fundamental. And hopefully those examples I've shown you illustrate just how we've evolved or potentially evolved to respond. So most of what I've been talking about so far, and particularly this idea that we've evolved a good response, is obviously helpful. Like, this is about how if you cut your finger, this is how you recover. But can these responses actually be harmful? Can there be too much of a good thing in terms of these responses, or can they go wrong in some way? So this is the model, again, that I've sort of been hammering home. You have an injury, damp release, damage response, and repair. And it's quite, I think, easy to understand how that might help you heal from a wound. And again, I think this is involving those same pathways that we think exist in really badly injured patients. So release of mitochondrial components, release of histones. And we've evolved to have these. But in my line of work, we look after patients who've had injuries that we haven't evolved to survive. Really serious injuries that, even with medical care at its absolute, with throwing everything at people, we still can't save people from injuries. And what we see in these people is actually, more and more we can actually get people through the first few hours. We can do an operation, we can stop the bleeding, we can get patients onto intensive care. But they have this massive immune response, basically. They have this massive inflammatory response, we think. They end up on intensive care, and they basically start getting failure of uninjured organs. So their lungs don't work, their kidneys don't work. And in some cases, we can support the patient through this, in which case they spend a lot of time on intensive care, they slowly get back to health. In some cases, we can't, and this is a major cause of death and disability. And we still don't fully understand why this happens, how you get from damage response, release of DAMPs, to a point that can actually become dysregulated. Obviously, we're doing a lot to these people. We're treating them with lots of blood, we're operating on them, so it gets very complicated quite quickly. But this idea of something being dysregulated when you're faced with a really big injury, I think there's also crossover with infection. And I'll talk about this again in a minute, but we're always getting and fighting off infections, but then sometimes your response can be dysregulated. It can be exaggerated, and that's what we call sepsis in bacterial infections. So I think this is kind of similar, but without an infection. That's how I would conceptualize it. So big injury, massive amounts of these damaged molecules being released, not just localized to a particular area, but actually all through the body. You get a dysregulated response, and this can culminate, we think, in this organ failure and even death. This is really the focus of our research. So how do these responses actually become harmful? What's actually happening to potentially cause harm? Well, I'll go back to this example of the histones. Again, those things that DNA is wrapped around that are present in the blood at really high levels. So these histones are actually, when you have them in high enough concentrations outside the blood, they're actually toxic as well to cells. This is a really interesting feature of histones that most other damaged molecules don't have. And this has been studied in quite a lot of detail. These are very, very fine resolution images of a cell membrane. The top one is just a control, so normal. And then this is with histone on it. And basically what you can see is that these are little holes in the cell membrane. So if you have a lot of histone around, it actually punches holes in the cell membrane. And again, this only happens when you have really, really high concentrations. If you have lower concentrations, it will activate cells through those receptors. But if you have really high concentrations, it will also start actually punching holes. And this I really like this graph. So these are two cells. This is over time in minutes, from just the start down to 10 minutes after exposure to histone. The green dye is basically is contained within cells that are intact, and then it leaks out of cells that are leaky. So it's disappearing because the cells are basically getting holes punched in them by the histone, and it leaks out. So thinking back to those pictures of cells bursting, jelly beans leaking out. And then the red is actually a marker of cell death. So cells that are dying will fluoresce red. So basically, within about 10 minutes of seeing the concentrations of histones that we find in the blood of our patients, the cells start actually dying. And we've done quite a lot of work on this. Others have done work on this showing that basically if you have lots of histone in the blood, it can actually cause damage to other tissues. So the idea here is really that, let's say you have a crush injury to your legs, you will release huge amounts of histone into the blood, and that histone is actually then damages other cells in, for example, your lungs, causing you to need to be ventilated. So this is the kind of thing that we think might drive that sort of problem. And what we're exploring is actually, can you block that? Because there's lots of molecules actually that will bind to histone and neutralize it. So can we actually give an injection at the roadside, something that the helicopter can take out, something that soldiers can take onto the battlefield, that can be injected at the scene, that can bind that histone, neutralize it, and stop the cellular damage. And we think this is quite a promising protective strategy, one that we're currently actively pursuing. And just to say as well that histones, this phenomenon of your cells leaking histones out actually happens in loads of other conditions. So it happens in sepsis, so dysregulated infection. It happens in coronary artery disease, atherosclerosis. Histone is thought to contribute to plaques being unstable. So it probably is a really fundamental mechanism of how your body figures out or thinks there's something wrong, going back to that danger model that Polly Matzinger described that I talked about at the top. So lots of crossover, I think, between what we see in our severely injured patients with other acute diseases, infections in particular. I think this is really interesting, and it's something that we're actively looking at more and more is, what are the lessons that we can learn from one condition to another condition, and how do these things overlap? A great example is COVID, where actually histones are strongly implicated in some of the responses that were observed. Lots of what we see in COVID clinically, in terms of massive inflammatory responses, similar in some ways to what we see in severely injured patients. So I think this is really about fundamental ways in which the body responds to damage. And I think by studying these patients, we can learn lessons that potentially help us understand lots of different diseases, as well as treat injured patients better. Okay, so last bit I want to talk about is the concept of why people might be different. Because what we see clinically, we have this model of cell damage, and certainly if you have lots and lots of cell damage, you're more likely to have problems than if you have very little cell damage. But actually clinically, we see some patients who have really severe injuries who seem to do absolutely fine. They just recover really quickly. We don't, in a way that we can't really predict very well. Whereas other patients seem to get this dysregulated, exaggerated response that I sort of talked about before, leading to failure and dysfunction in other organs. And we don't really understand this. We sometimes, on the ward, we'll scratch our heads about patients who have been right at death's door 1 minute, and then the next day they're sitting up talking to you. There's definitely a part of it is definitely how bad the injury is, but there does seem to be variability. And again, this is something that in infections we know that there is inter-individual variability, i.e. different people respond differently. So in trauma, we're still trying to get to grips with why this might be. This is a major focus for us. So there's a couple of, I think, targets for us to look at and try and understand that might explain this variability. The first is actually how these damaged molecules get out of the cells. This is probably a bit too simple, this idea of you bang your hand and then the cells just explode and they're physically disrupted. it may actually be that this is an active response. And then the last thing that I think is really interesting, I haven't really talked too much about, is your body actually also has evolved mechanisms to kind of mop up damps, too many damps. Yeah, so, and we all have these, but actually they may vary a lot between individuals. So some of these proteins that we're studying that neutralize histone, we may have very different levels of that protein in this room. So some of us may be better equipped to actually deal with and neutralize some of these toxins than others. And I think this is another way in which there might be variability. So lots of work still going on between the Royal London and the blizzard here. I would add that we've been at the forefront of a lot of this work. A lot of it's been done here. We've contributed to a lot of it. We still have a lot of trauma around, and we're still doing a lot of work to try and unpick some of those unanswered questions. So yeah, long road ahead, but thanks very much for listening, and happy to take questions.